Today we'll finish the rest part of the blood. First we'll talk about hemostasis. Hemostasis is the process of stoppage of bleeding or arrest of bleeding or haltage of bleeding. You know that when you get a cut, bleeding occurs, and after within few minutes, usually bleeding stops. And the process of stoppage of bleeding is called hemostasis. Now, hemostasis occurs in three mechanisms or steps. What are those three steps of hemostasis? First is vascular spasm, then platelet plug formation, and then blood clot or coagulation. So let's see what happens when you get a cut. So this is a blood vessel, and when you get a cut here, this is the lumen. So first thing what happens, you know that the wall of the blood vessel is smooth muscle. So contraction of the smooth muscle will cause narrowing of the lumen. So the size of the lumen will get smaller due to contraction of the smooth muscle in the wall. Okay, so that is what first happens. Then that is called the vascular spasm. It is like, the lumen size will get smaller. Then next what happens, platelets love the rough surface. So the platelets will start to aggregate and form a plug. So that will block the opening. So that is called platelet plug formation. Platelets love the rough surface, so some platelets will start to get attached to the rough surface of the blood vessel, and then more and more platelets will come and join to form a plug. So that is step number 2. Step 1 is vascular spasm due to the contraction of the smooth muscle, and that contraction occurs due to the pain reflex. When you get a cut, pain in your smooth muscle, you have nerve fibers, so that will cause pain, and that will cause the contraction of the smooth muscle. And then what happens, clot formation occurs. What is that? The fibrinogen, one type of plasma protein. Fibrinogen produce fibrin threads, and the fibrin threads will get attached to each other to form a net. So these are fibrin threads, and they will form a net, and that is the clot formation by the fibrinogen. So those are three steps of hemostasis or stoppage of bleeding. Now, what causes the vascular spasm or constriction of the lumen? When injury occurs, as I mentioned, that pain reflex occurs due to nerve fibers there, and that is the main reason why the contraction of the smooth muscle occurs. And here you see that aggregation of platelets. These are platelets, so platelet plaque formation, and this is vascular spasm. The lumen size will get smaller, and then coagulation or blood clot formation will occur by fibrinogens. So let me show you here again. So this is an artery, for example, and this is the lumen. So when you get cut, first what happens, the size of the lumen will get smaller due to the contraction of the smooth muscle, like this. So the size will get smaller. So the bleeding will be less. Then the platelets will get attached to each other to form a plug. So this is the platelet plug, and then fibrinogen, that plasma protein, will form a network that is called the fibrin mesh by the fibrin thread, and that will stabilize the platelet plug, so it will not get detached from there. So those are 3 steps of hemostasis. Now coagulation or blood clot formation. In your blood there are clotting factors, are also called clot factors in the blood. Those clot factors are also called coagulation factors. Okay, and there are total 13 coagulation factors, and we indicate those by Roman numbers. This is factor 1, 2, all the way factor 13. Okay, and all these factors are involved in blood clot formation. However, factor 1 to 4, that means first 4 factors, factors 1 to 4 are called the essential factors. Essential coagulation factors. Why those are called the essential factors? Because if any of these 4 factors is absent, then blood coagulation or blood clot formation will not occur at all. So you need to know those 4 important coagulation factors. What are those? Number 1 is that plasma protein. You have already learned about that, fibrinogen. Okay, so that is number 1. Number 2, factor number 2 is called prothrombin. Factor number 3 is called tissue factor. And factor number 4 is called calcium ion. Calcium ion. So remember these 4 factors. These are called the essential factors because they are essential for blood clot. Without any of these, blood clot will not be formed. Other factors, factor 5 to factor 13, those are also important for blood clot formation, but those are not essential. Lack of one or more of those factors will delay the clot formation. So the bleeding time will be prolonged, but eventually blood will get clot or clot will be formed. Now just know that first two, fibrinogens and prothrombin, these are synthesized in the liver, produced in the liver. So if someone has liver failure or liver disease, then the production of these two factors will be less, and in that case the blood clot formation will be uh, delayed. Uh, Tissue factor, this one comes from platelets and damaged or injured cells of the tissue in that area where you got injury or cut. So tissue, that's why I call tissue factors. Tissue factor comes from the platelets and damaged or injured cells. And calcium comes from the food, of course, and the bones of the body. You know that bones store calcium, and vitamin D also helps in calcium absorption. So those are the important factors or sources of calcium in the blood. Okay. Now there is another factor that is called factor number 8. Just know that factor 8 is called anti or anti-hemophilic factor. Its name is telling you it works against hemophilia. Hemophilia is a clinical condition. Hemophilia is a clinical condition in which bleeding occurs easily. So if you get a cut or small injury, lot of bleeding will occur and will continue for a longer time. That is hemophilia. So this factor 8, which is anti-hemophilic factor, that means it prevents bleeding or hemophilia. Now, how those factors, first 4 factors, get involved in clot formation? You see here, factor number 2 is prothrombin, right? So what happens? First, prothrombin is activated when you get a cut or injury. Prothrombin activator is formed, and that prothrombin activator, its name is prothrombin activator. So it will activate this factor number 2, prothrombin, and will convert it to thrombin. So it is telling you that prothrombin is an inactive. That's why it needs to be activated. So this is inactive. Okay, and it is converted to active thrombin. So thrombin is active. Who converts the inactive prothrombin to active thrombin? Prothrombin activator. Okay. So that is first. So first prothrombin activator is formed, then prothrombin activator converts inactive prothrombin to thrombin, active thrombin. Then thrombin will do what? Act on this factor number 1, fibrinogen, and will convert it to fibrin thread. You remember I said fibrin thread, and fibrin threads get attached to each other to form a fibrin mesh that covers the clot, platelet plug, and stabilize it. Now, how the prothrombin activator is formed? Remember, prothrombin activator is formed by two pathways. One is called the intrinsic, another is called the extrinsic. So intrinsic pathway and extrinsic pathway. You see here, intrinsic and extrinsic. Now, what will activate the intrinsic pathway? Intrinsic pathway is activated inside the blood vessel without injury to the tissue. What happens inside the blood? You have the platelets, right? So if platelets get injured inside the blood vessel, that will release the platelet factors. Okay, and platelet factors can also be released if the inner lining of the blood vessel is injured. That is called the endothelium, inner lining of the blood vessel, smooth lining. So platelet will release the platelet factors if injury occurs in the inner endothelium or platelet itself. And that platelet factors will activate or trigger the intrinsic pathway. So without any injury in the tissue. Now, if you get a cut from outside, that will injure the tissue, right? Your muscle, your wall, muscular, smooth muscle wall of the blood vessel will be injured. And that will release what? Tissue factor. And tissue factor will activate the or start the extrinsic pathway. So you see, two pathways are activated differently. However, finally, both pathways will produce a common factor that is called the prothrombin activator that I talked in last slide, right? Prothrombin activator. And a number of reactions occur in these two pathways, and you remember there are 13 clotting factors. Many factors are involved in these pathways. We don't need to discuss detail of that, just know that how they are activated, intrinsic pathway and extrinsic pathway, and finally they form same thing, prothrombin activator. And then prothrombin activator, you see here, has been produced both extrinsic and intrinsic pathways. And prothrombin activator, as I have mentioned, will do what? Its name is prothrombin activator, so it will activate the prothrombin, which is inactive to active thrombin. I wrote it in last slide. So this is active, this is inactive. Then this active thrombin will convert the plasma protein fibrinogen to fibrin. And fibrin is thread-like, string-like structures. So many fibrins will be formed and they will form a mesh network that is called fibrin mesh. Okay, and you remember I talked about the hemostasis, arrest of bleeding. So platelet plug is formed here. Remember that, to block the opening here. And then this fibrin mesh will cover this to stabilize that platelet plug there. So that's how the bleeding stops. Now you remember factor number 4, which is an essential factor, calcium ion, that is used in many reactions. In different reactions, the calcium is used also here. So calcium is a very important coagulation factor, and if you can inactive the calcium in your blood, then blood clot will not be formed. Sometimes when we collect the blood for lab test, we want to keep the blood liquid for hours, so we remove the calcium from the blood by calcium chelating agent. So that will remove the calcium, or take off the calcium from the blood, so blood will remain liquid. Here you see some red blood cells and fibrin mesh. It's like a net. After the clot is formed, retraction of clot starts, and that retraction occurs between 30 to 60 minutes after the clot is formed. And why clot retraction occurs? You know that platelet plug is formed by the aggregation of platelets, right? And so this is the clot covered by fibrin mesh, right? And what happens inside the platelets, you have actin, myosin, contractile proteins. You have learned about actin, myosin in NP1. So actin myosins will do what? Will contract the platelets. So the size of the platelets will get smaller, and that will squeeze the whole clot and will make it smaller. So that is the clot retraction, squeezing of the clot. And when that will happen, clot is a semi-solid structure, so it has fluid inside it. So that fluid will come out, and that is called the serum. So this is the serum. Serum comes out when clot retraction occurs. And remember, serum is actually plasma, the liquid part of the blood, minus clot factors. Why is that? When clot is formed here, the clot factors have been already used up, right, to form the clot. So clot factors of the plasma of the blood has been used to form the clot. So the liquid that remains and comes out, that one is plasma minus clot factors, and that is called the serum. So plasma has clot factors and serum has lack of clot factors. Disorders of hemostasis. There are two types of disorders we see. One is called thromboembolic disorders and another is bleeding disorders. Thromboembolic disorder is if any clot is formed inside the blood vessel. For example, this is an artery and a clot has been formed inside the artery. That can occur due to intrinsic factor activation, you remember, without any external injury. And that clot will stay attached to the wall. So it is fixed, and this type of clot inside the blood vessel is called a thrombus. So the clot is attached to the wall. Now sometimes what can happen due to the flow of blood, this clot may get detached from the wall. So now this is a floating, freely moving or floating clot in the blood, and this one is called an embolus. Okay, embolus. Now embolus is more dangerous than the thrombus. Why? Because this embolus will now move inside the circulatory system or inside the blood vessels, and from here it can go to the heart or to the brain, and you know those are two very important organs, most important organs of your body, and can block the artery of the heart or the artery of your brain. So this freely floating clot, which is called the embolus, could be very dangerous, can go to the brain and block the artery of the brain or coronary artery of the heart. That can cause heart attack or stroke. So that can cause stroke in the brain or heart attack or myocardial infarction. Which is also called heart attack in common term. Infarct, R-C-T-I-O-N, M-I, probably you have heard that. Myocardial infarction is M-I, or heart attack. Okay, so that's why abnormal clot formation is inside the blood vessel, which is highly undesirable. We don't expect or desire any clot formation inside our blood vessels, and that is thromboembolic condition. Now, bleeding disorders, I already explained this. You have 13 clotting factors or coagulation factors, and if any factor or factors are absent, that can cause prolonged bleeding. The clot formation will be delayed, and in that case, the bleeding will be prolonged. Now, undesirable clot formation inside the blood vessel can be prevented by some chemicals. One is aspirin. Aspirin uh, inhibits thromboxane A2. You must remember in last lecture I mentioned that platelets uh, get sticky due to thromboxane A2, and another thing I mentioned, ADP. And these two chemicals make the platelets sticky. So the platelets can easily get attached to each other to form a clot. So aspirin inhibits the thromboxane A2. So the platelets will not easily get attached to each other. So that can prevent the undesirable clot formation or thromboembolic condition in the blood vessels. Heparin is a natural anticoagulant. I mentioned before, natural anticoagulant. So it keeps the blood thin, prevents the coagulation. And why it is called natural? Because you know that white blood cell basophil, I mentioned before, basophil secretes the heparin. So those two. And there is another chemical there that is called warfarin. Warfarin is also an anticoagulant, and this anticoagulant is also called blood thinner and can prevent the abnormal clot formation uh, inside the blood vessel. Blood group or blood type. You know that different people have different blood group or blood type. Why is that? Blood group is actually determined by the surface of the red blood cell of that person. So this is a red blood cell, okay, and on the surface of the red blood cell you have proteins called antigens. So what kind of protein is present or absent, that determines the blood type. So if on the surface of the red blood cells you have antigen A, your blood type is A. So this is type A blood. If someone's red blood cell surface, this is hemoglobin, uh, different types of antigens, like this is antigen B, antigen B. So this person's blood type is B. So blood type is B. Okay. And if someone's red blood cell surface has both A and B, that means this blood type or blood group is what? AB, because this one has both antigen A and antigen B. So this blood type is what? AB. And the last one, if someone's red blood cell so it's let's go back here. So if someone's this is another red blood cell, and on this one no A antigen, no B antigen, neither A nor B. So this person's blood type is what? O. So what type of antigen is present on the surface of the red blood cell, that determines the blood type. If A is present, type is A. B antigen is present, type B. A and B both are present, blood type is AB. None of those, blood type is O. Now, there are some other antigens, those are very weak, so they don't play any role in blood typing. For example, MNS, Duffy, Kell, Lewis, Lutheran, those are some weak antigens present on the red blood cell, but we don't consider them as a determinant of blood type. So A, B, those are the antigens. Now, there is another antigen that determines if the blood type is positive or negative. Okay, so you know that some blood types are positive and some are negative. So who determines that? That is determined by another antigen that is called D. So if someone has D antigen, that is positive. If D is absent, that is negative. Okay, so now you just think that this is a red blood cell, hemoglobin, and this person has A antigen and as well as antigen D. So this is A and this is D. So what will be the blood type? Because of A, the person's blood type is A, and because of D, the type is what? Positive. So this person is A positive. If another person, he doesn't have A, he has B antigen on the surface. So this is B, and also he has D. So his blood type is B positive. Now if D is absent, only B, blood type is B negative. B negative. Okay. If this one is absent, A negative. If someone has no A, no B, no D, that means O negative. Okay, so that is how the blood group is determined by the presence or absence of those antigens. Now another thing which is very important to remember, if on the surface of the red blood cell any antigen is present, for example this one has antigen A on the surface, outside of the blood cell you have the plasma, right, the liquid part of the blood. So you have blood cells and plasma, so this is plasma. So this is very important to know that if any antigen is present on the surface of the red blood cell, same type of antibody must be absent. So same type, this is a rule, antibody. So in the plasma you have antibody must be absent, where in the plasma. That's a rule. Okay, so on the surface of the red blood cell you have antigen, and in the plasma you have antibody. Now if on the surface you have a antigen, in the plasma you must not have that same type of antibody. If antibody, you have same type of antibody, then antigen antibody reaction will destroy the cell. That is very important. Okay, so that's why same type of antibody must be absent. But who can be present? The opposite type antibody. So they will not react to each other. So if on the surface you have A antigen, that means blood type is A, right? In the plasma you have B antibody. Is it clear? Antigens are also called agglutinogens, and antibodies are also called agglutinins. Okay, now let's again go back. This is blood type A, type A. Okay, that means on the surface you have A antigen. Okay. And in the plasma outside of the cell, in this case you have opposite antibody, not the same type, B antibodies. Okay, so this is type A. And now let's draw another red blood cell. On the surface you have B antigens. Okay, so type B, blood type is B. Okay, and in the plasma, in this blood, a type of antibodies should be present. The opposite, same type is dangerous, will never be present. Okay, corresponding antibody must be absent. So this one has A antibodies. Okay, now let's see if this person, this is donor and this is recipient. Okay, So this person is giving blood or donating blood to this person. Now remember, this is another rule. Rule number 2. When blood transfusion, uh, uh, we donate blood to someone or blood transfusion occurs, we consider the red blood cell of the donor and the plasma of the recipient. Okay, so we consider these two things. The red blood cells of the donor, we don't consider the plasma of the donor, only the red blood cells of the donor, and plasma of the recipient, not the red blood cells of the recipient. Okay, so now let's see. This person is donor, so we'll consider his red blood cell. On the surface of the red blood cell, this person has A antigens, right? So when this red blood cell will enter into this person's body, recipient's body, you see in the plasma of the recipient of B blood, you have huge amount of A antibodies, right? Because in your blood you have 55% plasma, right? So plenty of A antibodies. So when this red blood cell will enter into this recipient's body, in his plasma you have huge amount of A antibodies, and these antibodies will destroy the red blood cell of the donor, because on the surface you have same type of antigen, right? So this antibody will destroy the red blood cell, will get attached to the antigens here and will destroy or cause the lysis of the red blood cell. So when this blood will enter into this person's body, all these red blood cells of the donor will be destroyed by the same type of antibodies. Make sense? If this B type blood is given to type A, now this is the donor and this is the recipient, right? Opposite. Same thing. On the surface of this red blood cell has B antigen, and in the plasma of this blood has B antibodies. So the donor's red blood cells will be destroyed when it will go to this person's body, because the antibodies, same type of antibodies, will destroy the membrane of the red blood cell. That is called agglutination. The process how antigen antibody reaction will destroy the red blood cells. Okay, now let's think another thing. This is O blood, right? Blood type O, and this is blood type A. So this person's red blood cell surface, you have A antigens, right? In the plasma you have B antibody, right? So this is type A, and type O has no antigen on the surface, no. So you see, when this blood, O blood, will be given to type A person, then what will happen? this red blood cell will not be destroyed because on the surface there is no antigen. So this antibody, any whatever the antibody is, in this case antibody B, right, will not be able to get attached on the membrane because no antigen is in O. So O can be given to any other blood. So that is why O is called the universal donor. Even you see, if this person is B, on the surface this person has B antigens, and in the plasma this person has A antibody. But since this red blood cell has no antigen, this one A antibody will not be able to destroy the membrane. So O can be given to any other blood. The last thing, AB. AB is universal recipient. Why? You see, this is AB. That means this red blood cell surface, you have both A and B. So A and B antigen, right? So corresponding antibody must be absent. So in the plasma of AB, no antibody. Because on the red blood cell you have both antigens A and B. That's why it is type AB. So in the plasma, no A, no B antibody. So whatever the blood goes to this blood, if this is A, for example, on the surface of these red blood cells you have A, right? But since in the plasma no antibodies, so when this red blood cell will enter, since no antibodies, they will not be, nothing will get attached here, right? So it is safe. B can also go there. You have B and antigens here, but since no antibodies in the plasma, any type of blood can be given to AB. That's why AB is called what? Universal recipient. Recipient. Okay, so O is universal donor, AB is universal recipient, and you can practice at your home by drawing different types of blood, antigen, antibody, and uh, practice that which one uh, will cause transfusion reaction, which ones will not. So now we have learned that ABO, this is one system that is called ABO system or type, that includes type blood type A, B, O, AB, right? So these four blood types belong to ABO system and determined by antigen A and B, their presence or absence. And another is RH system, that is positive or negative, whether blood type is positive or negative. That is determined by what? Antigen D. D antigen determines that. Okay, if D is present, positive, D is absent, negative. And that is called RH system. Why? Because this D antigen was first isolated in rhesus monkey. So that's why this name was given RH system or factor. A clinical condition that is called erythroblastosis fetalis. Erythro, red blood, right? Cells. So red cells, and blast, erythroblasts, young, immature. Cyte means mature, right? And blast, young. You already probably know. So erythroblasts are young red blood cells. Fetalis in fetus blood. So if in fetus blood you find plenty of immature red blood cells, that is called erythroblastosis fetalis. It is a clinical condition. Fetus may suffer from this clinical condition, in which in the blood, instead of mature red blood cells, you have huge amount of immature red blood cells. And you must remember, immature red blood cells have nucleus but no hemoglobin, right? When maturation occurs, nucleus is kicked out and hemoglobin appears. So this immature red blood cell is non-functional, cannot carry oxygen, because to carry oxygen you must have to have hemoglobin inside. Hemoglobin binds with oxygen, right? So although the fetus blood has a lot of red blood cells, but those are immature and have no hemoglobin but nucleus, so cannot carry oxygen. So that is a big problem. Now, why that happens? That happens if the mother is negative, Rh negative. Mother's blood is negative and the fetus is Rh positive. Okay, Rh positive. In that case, what happens, positive means on the red blood cell surface you have D antigens, right? You already know that. And in mother's blood, red blood cell doesn't have D antigen. So this is fetus blood, and fetus blood has D antigen. Now when the fetus blood goes to the mother's body from the uterus, you know that blood circulates from the fetus to the mother. So this D antigens enter into the mother's body. And just know that when any antigen enters into someone's body, the body responds by producing antibody against it. Like microorganisms, those are also antigens, right? Or the D antigens. So when D antigens enter into the mother's body, in response to that, it is a natural response, the mother's body will produce D antibodies against it. That is a natural response. So the D antibodies will start to produce in the mother's body. Now when before plenty of D antibodies are developed in mother's body, first child or first birth occurs already. So the first child or newborn usually don't suffer from this problem. But when the same mother will get another Rh positive fetus, will get pregnant again with Rh positive fetus, then what will happen? Again, D antigens will enter into the mother's body, and this time mother's body or blood will produce plenty of antibodies quickly, very fast, because it is already sensitized, it knows. So in this case, mother's body has already a lot of antibodies, because after first birth still mother's body continue to produce antibodies, and when second time antigens will enter from second fetus, then it will produce more antibodies. So that antibodies will do what? Will go to the fetus body. So now the fetus red blood cells, since the blood type is positive, whatever positive, it could be AB or AB positive or O positive, on the surface, since positive on the surface you have D antigens, right? That makes positive. So this is fetus blood, red blood cells. Okay, and from mother's body plenty of D antibodies will come. Now will enter into the fetus body, and these D antibodies will react with the D antigens on the surface, right? And will destroy, same type, so will destroy the red blood cells of the fetus. So all these red blood cells will be destroyed. So when the red blood cells of the fetus will be destroyed, then the bone marrow or the site where the red blood cells are produced, liver and bone marrow, will do what? Will try to quickly replace, since in the circulation the number of red blood cell is now very low because of the distraction. So the liver and bone marrow will quickly try to replace by producing new red blood cells. But most of these red blood cells are immature because they are being produced very fast and given to the blood circulation. So these immature red blood cells cannot carry oxygen that I mentioned before, because hemoglobin is not yet inside these immature red blood cells. So that's why this clinical condition is called erythroblast, immature red blood cells fetalis, in the fetus, and those are uh, incapable of carrying oxygen. So those are not really functional red blood cells. In subsequent pregnancies, the chance of erythroblastosis fetalis increases. In third pregnancy or fourth pregnancy, the chance is even more. Now, what happens in erythroblastosis fetalis? You already know that the destruction of red blood cell will do what? Will release the hemoglobin, and hemoglobin is converted to yellow pigment called bilirubin. You have already learned about that, and that may cause jaundice in the fetus, right? Newborn. But this jaundice is not physiological jaundice that we often see in the newborn baby. Okay, this one is due to uh, blood type, a negative blood type of mother and fetus positive. And now since now we know the reason why the fetus gets erythroblastosis fetalis and fetus die um, due to this clinical condition, we now know how to prevent it. Before the mother's body gets D antigens from the fetus blood, if you know that the fetus is going to be positive, you can desensitize uh, before the second pregnancy. So if you know that first child was positive, that means in mother's body already D antibodies have been developed, right? So uh, if mother is Rh negative, then you will take precaution. You will uh, inject or give the drug, a medicine that will inactivate the D antibodies or production of T antibodies. So in that way, you can prevent the erythroblastosis fetalis in subsequent uh, pregnancies. So